Okay, so structural biology. Structural biology is a field of biochemistry that uses a variety of techniques to determine the 3D structure of biomolecules such as proteins, nucleic acids, and their complexes. So generally in structural biology, there are a variety of techniques that are used to know the 3D structure of a biomolecules which help in identifying their functions. So structural biology is one of the great trending area of research currently that are used in elucidating the structure of biomolecules. So the structure of biomolecules can be looked at different levels. It can be start from atoms, molecular levels, and then the composite levels. We know that biomolecules are started actually from atoms, then from atoms to molecules. Even at the molecules levels, it can be start from simple molecules to complex molecules. And these complex molecules or macromolecules can start from proteins, nucleic acids, and of course, these macromolecules form a supramolecular complexes. So these techniques allow researchers to elucidate at different resolution from atomic to supramolecular levels. So structural biology focuses on the study of biomolecules, interactions, and dynamics, that is, how biomolecules interact and change over time. So structural biology uses a technique to study the architecture of molecules at different resolution ranging from atomic level to supramolecular complexes. And of course, the structural biology is focuses in trying to know the biomolecular interaction and their dynamics, as well as how the biomolecules interact and how they are changed over time after the interaction. Like for example, the interaction between proteins and ligands. Structural biology is important from understanding the functions of biomolecules and their roles in health and diseases. Like, for example, there are so many molecules that are associated in our health and diseases. So, structural biology will help us to understand how the structure and the functions of the biomolecules are associated with health and disease condition. And there are various techniques that are used in structural biology, and we are going to look at some of them in brief. So this is actually a typical examples of X-ray crystallography techniques. So like for example, in this X-ray, when you have your crystal, then the X-ray will be actually applied and the structure will be elucidated. So advances in computational method and techniques have also played a significant role in structural biology. This helps to provide new ways to analyze, interpret, and integrate data from different techniques, thereby enable researchers to have a deeper understanding of biomolecules, dynamics, and relationship to biological processes. So due to advances in computer method and techniques, it now helps in interpretation, integration of data from different techniques, and this helps to researchers to enable them for deeper understanding of biomolecules, their dynamics, and the relationship to biological processes. So it means that, so number one, let's look at the X-ray cryo-electron microscope. Cryo-electron microscope is used in structural biology It's used in structural study of proteins, especially those that cannot be studied by other techniques such as large protein complexes, membrane proteins, or proteins that cannot form crystal. So in cryo-electron microscope, it's types of electron microscopy that are used in studying larger proteins, which include membrane proteins and some of those proteins that cannot actually be studied using, or that cannot form a crystal. So if they cannot form a crystal, you cannot use that. Then we have X-ray crystallography. This technique is based on the fact that a crystalline sample is able to cause the diffraction of an incident X-ray beam, generating a characteristic pattern that can be used to deduce the structure of the crystal at an atomic level. So the crystalline sample is irradiated with X-ray beam at different angles of incidence. So in X-ray crystallography is the X-ray diffraction patterns that are used in deducing the structure of the crystal at an atomic level, and that would lead to the elucidation of the structure of the biomolecules. So you see, in this case, we are using X-ray diffraction pattern of the molecules and then employ that in understanding the 3D structure. The NMR spectroscopy is also a technique that take advantages of physical phenomenon in which the atomic nuclei are irradiated with radio frequency and reach excited energy states. And these techniques provide information like structural elucidation of macromolecules, study of macromolecular dynamics, and then characteristics mechanisms of the interaction between biomolecules. So NMR spectroscopy can be used actually to elucidate the structure of biomolecules or macromolecules like proteins, nucleic acid, and also provide information of the dynamics of the molecule as well as the mechanisms of the interaction between the biomolecules, like for example